Hello, everyone. I'm Purnima. I'm the product manager at Krishagni, and I have with me Shrikant Adiga, who is the CEO. So today we'll be giving you an introduction about OpenSpecimen, different features and capabilities. If you are interested in specific one-on-one demo, we'll be happy to do it based on your use cases. So first we'll start with some introduction about us and about the product, but we'll spend most of the time showing you the actual features of OpenSpecimen. We have been working with various biorepositories, research centers, hospitals across the globe since 2009. And we are now working with about 90 plus biobanks, research groups across different 20 plus countries. Some of the big universities like Washington University, St. Louis, Jefferson, that is the Thomas Jefferson University, UPMC in Pittsburgh, Columbia University, all of these have been working with us for more than 10 plus years, and they use it at enterprise level, that is for the whole universities, which mean that they use it for multiple groups, multiple research groups within the institution. For example, Washington University uses it for more than 30 plus labs, multiple hundreds of protocols, hundreds of users using concurrently for millions of samples. So it is used both at large scale like this, as well as individual PIs or smaller clinical studies. So there is no restriction on how big the installation is or how many users are going to use it. So there is no restriction in terms of licensing. So yeah, we already talked about this. As far as our company is concerned, we are solely focused on the product development of OpenSpecimen, development of new features, helping our clients implement OpenSpecimen in terms of installation, training, legacy data migration. We help our customers to migrate their data from Excel sheets, in-house databases, and so on. And also if they need any new features, enhancements, integrations with other systems, all of these is what we work on. And so now going into the features of OpenSpecimen, it allows you to track the whole life cycle of the sample, starting from collection, processing, storage, utilization. So everything that happens to the specimen during its life cycle, it can be captured in OpenSpecimen. So if you talk about the typical biobank, as you see in the diagram, this would be the workflow that you first start with the patient registration, coming into the hospital for consent, giving the samples where you collect, the clinicians will be collecting any demographic data, clinical data. So they might either collect this data directly into OpenSpecimen, or if they're already using any hospital systems or REDCap, they can collect that and that data can be transferred over into OpenSpecimen. Now the specimens arrive in the lab where the specimen processing happens at the lab or the biobank, and the processing, any QA or any tests that they perform, and they might store it for long-term storage in their freezers. Now, the data can also be exposed to your researchers where they can look for samples, request for samples, and finally, you can process that request to distribute, that is the utilization part of the specimens. So as you see, every step of the sample can be captured. Again, it's not mandatory that you do all of these in OpenSpecimen. It's all based on your requirement, your workflow. So you can pick and choose what you want to do in OpenSpecimen, which we'll be seeing in the demonstration. So now, what types of collections are supported? So prospective collections like disease-based or biorepository, general biobanking collections, longitudinal collections. So typically you might have clinical trials where you're collecting samples across multiple time points, like baseline, screening event, there is a day 30 follow-up, surgery event, follow-up after surgery. So these kind of time points or study calendar can be defined within OpenSpecimen, and you would be able to collect samples across these time points for specimen collection, processing, and so on. You can even do multi-site studies. So again, the scenario would be there are different collection hospitals where the collections are happening. They then send the samples to the processing lab, storage facility. So all this collaboration between multiple groups. It might be within your institute, across institute, across universities, across US, or even across the globe. There is flexibility to do any kind of study. So OpenSpecimen is a web-based application. Now, who has access to your instance is totally dependent on how you install it. So some of our clients make it publicly accessible. So although the backend security handles all the security of the data, so the URL would be publicly accessible so that users across the globe can access. Or if you want, you can just install it within your network so that only users within your university can access it. So there are, again, multiple ways to install the application. Again, it is left to your choice. We can also host it for you on the cloud. That is also an option. The system is highly configurable such that even you can do non-human collection. Some of our clients do mouse sample collections. Recently, we have been working with a group where they're doing gorilla research and they want to collect the samples and store it. And when I say configurable, it's not just the data fields that you capture. You can also capture or configure how you can do the data entry. That is, the data entry screens can also be configured. User roles can be controlled, who has access to what data in the system. And when you install OpenSpecimen, it comes with a default data model, but you can extend the data model easily based on your requirements. You can apply complex data validation rules. You can manage request and distribution, that is the researchers themselves requesting the sample within OpenSpecimen rather than they calling you and asking for samples. One of the major highlights of OpenSpecimen is its reporting tool. It's very easy to use. Even researchers or daily users like tissue bankers can generate reports, get the data out easily from OpenSpecimen. You can import data in bulk using comma-separated value file. This is used by us extensively when we do legacy data migrations. We also have a separate mobile tablet app, which can be used to do any kind of data entry. This is especially useful for offline data entry. For example, you have gone to offsite collections where you might not have access to internet. You can use this. Of course, it does not mean that you cannot use within internet. You can use it anywhere easily. This is more for ease of use. We make sure that the performance is not, is up to date. For example, any operation should happen within 5 seconds. So the scalability is taken care of in terms of how many users are using. So there is no restriction on how many concurrent users are using, how many samples are stored. So there is no performance issues in those cases. We make releases every 3 to 4 months. This includes bug fixes, enhancements, features, new features. This is all based on community feedback. One is when we work with new clients, if there are any gaps they come up with, then that would be part of the new releases. Or our existing customers give continuous feedback and we prioritize them and include in the releases. This does not mean that you have to update every new release. It's left to you. You can choose which release to go for. So we do have public forums. So we have forums.openspecimen.org where we have different announcements, product features that we keep doing. So if you see, we are hosting some workshops which are coming up in London and US, and any new releases we make, we post it on this forum along with the release notes. So every release notes has detailed description of what has been implemented in that release. So people can subscribe to this forum to keep up to date with the product. And this is our public manuals or help center where we have user manuals, deployment guides, and so on. And our user manual is also extensive in terms of every module has, all the steps in text-based, as well as there are videos to, guide the users to go through different modules of the application. So these are some of the resources that would be helpful for you to evaluate. And coming to the community feedback, we keep doing webinars like this on a monthly basis for all different features, introductory webinars, and so on. We do yearly workshops, community meets. Couple of the community meets which are coming up is one is in London and one is in US. As you see here, a lot of users from different centers, whether they're new users or existing users, they come together. It's more useful for networking, understanding how they're using, as well as we conduct a lot of workshops on new features, training, and so on. So these are some of the resources, again, that would be useful for your evaluation. Coming back to features, the system has 100% audit rate, which means that every operation that the user does is audited and super admins can generate reports for it. And finally, OpenSpecimen is completely based on REST APIs, so which would mean that you can do integrations very easily. So we have done a lot of integrations for our clients with applications, instruments, for example, clinical data systems like RedCap, OpenClinica. If you want to pull patient data, forms data, any kind of clinical data, you can do it easily, automatically, so that there is no duplicate data entry and so on. Similarly, we have done a lot of integrations with Epic to to pull in the demographics, consent data automatically. Not only software systems, we can also integrate with instruments, automated freezers. We have done integrations with Hamilton BIOS and other freezers, wherein again the messages sent by the freezers are consumed by OpenSpecimen, and automatically the storage data is stored in OpenSpecimen. These are again just examples, but if you have similar integrations, we'll be able to help you do the integrations with OpenSpecimen. So that's some of the introduction. So let's now dive into the actual demo of the software. Again, this is another very useful resource, that is our public demo site. It's demo.openspecimen.org, where you can create like a sandbox for yourself to go through different features of the application. We always have the latest version of the software deployed, so you will be able to get hold or get to see all new features. So recently we released 10.1. So this is the home page of the application. So as you see, there are different wizards, or you can go access through and go to different modules. Or on the left hand side, there is a navigation menu where you can access different parts of the application. So now what I see in the system, it completely depends on what role I have. So right now I have logged in as a super admin, which is the highest role. But there are different levels of access that you can give to a user. So there is an administrator who has access to everything. As you see in the permissions, you can control, read, create, update, delete, export, import, and so on. And these are all the resources or modules. So this will decide what they can do in the system, what they can see in the system. Even the user interface is driven based on this user roles. Can they see the delete button? That depends whether they have delete access or not. So you will be able to control the user access. As you see, there are some default roles in the application, but you can create your own custom roles. As you see, like if you see technician, this is more restricted role. The other thing about roles is you can also control who can see participant identified data. So if you don't want certain users to see things like participant names, SSN, MRN, those PHI kind of data, then you can control who has access only to de-identified data versus who has access to the PHI data. So this way, the roles are very flexible. You can control at a granular level what each user can do. So now once you have set up the roles, then you set up the users. So you can create any user with specific details. You can also integrate users with single sign-on, like if you want to log in with your university login, you can integrate with LDAP, SAML, and so on. Now this particular user, you can give them roles. So here if you see, this particular user belongs to Boston University. Within the Boston University, you might have different groups, like there might be lung cancer group, diabetes group, and these can be created as sites in OpenSpecimen. So site is nothing but wherever the specimen is handled, like, the collection hospital, repositories, they can be created as site, and each user belongs to a site. Now that will define what data they have access to. So OpenSpecimen can have multiple sites, and each site user can belong to one or more site. Within the site, you can have one or more protocols. So protocol is nothing but, the clinical study or the general biobanking protocol. So the collection of samples for a particular purpose can be categorized as a protocol. So now the combination of site and protocol, you can give them a role. So this particular user is a technician for this particular site or, university or biobank. Within that biobank, right now, this user only has access to this particular protocol. You can also give access to all protocols if you want. That is an option you have. So this way you can have multiple users. There is no restriction on how many users you can have in the system. You can give them different levels of access and so on. That's the users and roles. Now let's go into the containers. So containers is nothing but the freezers. So this is where you would be handling the storage of the specimens. Where are you going to store your specimens? Are they liquid nitrogen tanks, -80 freezers, even automated freezers, or even where you store the archival tissues like cabinets? So all those can be created as containers. So now how do you create them? You create it exactly how it is physically, structured. How many shelves does the freezer have? So this particular freezer, this is an example of -80 freezer having 4 shelves. Now you can see it graphically like this, as if you're opening the freezer. So now you have 4 shelves. When you see the first shelf, there are 6 racks. Now you can drill down to see the racks. Within the racks, you have then the boxes. Then within the box, you have, the specimens. So this particular box is 9 by 9, but you can have 10 by 10, 8 by 12. So any kind of box, any kind of rack. Again, this is just an example of the freezer hierarchy or structure, but you can define your own freezers the way you want. So here, if you see this gives, when you click on a particular specimen, it will give you more details about it. You can see the nice color coding here. This will indicate different types of samples. If you see here, the view color coding, it will give you what kind of samples are stored. So you can, set up your freezer and get an overall view of what kind of specimens are stored in the freezer, and you can, easily see where the specimens are stored. You can also see the specimens as a list view if you want. This is like a list view for each of the box or rack and so on. You can do different actions right from here. So this is the freezer view where you can access the specimens within the freezer. You can generate different reports of the freezer, how the freezers are utilized, each box, how many specimens are stored versus how many are free. So you can generate a lot of such reports. You can see graphs to get an indication of how the freezers are utilized. And setting up these freezers is something that an administrator can do through user interface. Someone like a user within your biobank will be able to do it. You don't need any IT support to do it. You can create multiple freezers. And if you see, each freezer is also associated with a site, which means that only these site users can access these freezers. So imagine a university has multiple biobanks. They have their own freezers. They want to work independently of each other. So this will allow you to, track your specimens and not give access to your freezers to other sites and so on. So the next part of the application is the collection protocol. So as I was mentioning, this would allow you to track your specimens based on different studies, different categories of the collections and so on. So if you see now, this is the demo site. So there are multiple protocols which someone has built. But if you see, there are some examples that I'm going to show today. One is the longitudinal study. So this is an example where this particular study is associated to multiple sites, multiple groups within your institute or outside institute. who is the PI, when is the start date, what is the IRB ID. If there is any SOP document, you can attach to it. You can also build like a study calendar if you want. This is like you're predefining what you're collecting, how you're processing the samples. So you can set up like a study calendar in advance. This will allow you to do the data entry easily. So you don't have to repeat all this for every specimen that you collect. So there is baseline where I'm collecting, 4 tubes of blood, 1 tube of urine. They're processed into plasma aliquots, buffy, serum aliquots, urine aliquots. So I have predefined like a template for my study, what I plan to collect, how am I going to process it. So this will kind of give you like a SOP of what needs to be done for this protocol at each time point. This is an optional thing. If you have studies where you don't know these details, you would be able to not define this and still collect any kind of specimens as you get the samples. So this is one option you have. You can also configure consents if you want. In OpenSpecimen, you're allowed to track consents, either like statements like this. You can attach consents PDFs if you want. And there is a separate e-consenting module as well, where you can let the patient enter the consent and e-sign the consent as well. So now when I go inside the protocol, I can see the list of patients or participants. Then I can go inside the participant to see, let me go inside a patient which has some data over here. So here, if you see, this has some demographics like gender, race, ethnicity, first name, last name. So these are the default demographic information which is present in OpenSpecimen. But this doesn't mean that you have to follow this and use all of this. You can hide the fields that you don't need. Every study can have its own data fields. So if you don't need certain fields for a study, you can hide them. You can add new fields easily for a study. There is a forms builder, forms module where you can add new fields easily. Then within each participant, you can track the different time points, different visits that the patient is coming in for specimen collection. So here is where you can also track consent. As I was saying, you can upload the signed consent form. And if you want, you can also track into the level of, the consent statement, whether the patient said yes, no, and so on. How this would be useful is then you'll be able to do reporting based on it. Give me all specimens where, the patient has said yes to process DNA, so that you know that you can process the DNA. So you'll be able to track all this. You can validate these consent when you distribute the specimens and so on. So now for this patient, if I want to collect specimens, I can go inside any of the visit and mark the specimens as collected. If you see these are already collected, I can go into the post-surgery. If I want to collect these specimens, I just select them and mark it as collected. So this is if I am doing as per my protocol, but if there are deviations, I will be able to mark them. Suppose I didn't make 5 aliquots, I made only 4, I can change that. I can track who is collecting it, at what date and time, when did they receive it, what is the quality of the specimen, where is it getting stored. When I process the aliquots, even the system can tell you or auto allocate what is the next best location for storage of the specimens. So you can even auto allocate. Of course, you can also manually change the container details and submit this. You can print the label formats if you want. You can print labels from OpenSpecimen, and any additional details if you want, you can capture like centrifugation, freeze time. So all these fields that you see, some of them are by default present in OpenSpecimen, but you can configure some fields based on your requirements per study basis. So there is also features which is called forms, where you can integrate with your clinical systems, where, for example, from RedCap, you can pull in the instruments data, which can appear automatically in OpenSpecimen, or you can build such forms directly in OpenSpecimen, where you will be able to add the data here, and enter any data that you want. So this is someone has built some sample form that they wanted to capture for patient, like a questionnaire. So this is, again, this form by default is not in OpenSpecimen, but there is a form builder. So there is a feature where you can create new forms. So this is to extend the data model. That is, if any data field is missing in OpenSpecimen, again, you don't have to ask your IT team to add that new field. As an administrator, you will be able to create a new form, add fields as required. For example, let me just go into fecal isolation. Here, if you see the quantity, G-force, time, user. So you can create any type of field like text field, text area, radio button. So any kind of new field that you want to capture for a study, you can easily add them and capture it at the participant level, visit, or specimen level. Yeah. So this is an example of a longitudinal study. Now, if you see, there are other examples. So suppose you don't want to collect any patient data. Imagine you're doing bacterial isolates, cell lines. In those cases, if you see here, we call this protocol as a specimen-centric protocol, where you don't collect any data about patient visits, directly collect specimen data. And here, if you see, the fields are completely different compared to the other study that we looked at. There is, fields like taxon name, the fields like, which is relevant to the bacterial isolates, right? So there is culture, there is other details that you can capture. So this is just again, an example that each study can look different. And the study definition, the study fields, how you capture the data, everything can be configured at the study level. Even in terms of, suppose you want to do any non-human collections. If you see here, now instead of patient ID, participant name, birth date, and so on, you have different set of fields here like animal ID, species. So within single instance of OpenSpecimen, you can collect any type of data, any type of study collections can be done. Each study can look different, can be configured the way you want based on what data fields you can capture. Now, all this data you can also import in bulk. As I mentioned before, you can do bulk imports through comma separated value file for any data like patients, consents, form data, events data, and so on. And now when you want to upload the data, there is a download template file. So you need to upload the data in specific formats, and it should follow all the validations like, mandatory fields, drop-down lists, and so on. So now once you upload the data, it would show all the view your past imports. Like if there is any failure, it will show you what the errors are. So you can track how the specimens are being uploaded. So this, as I said, we use it during the legacy data migration, but you can also use it for, future data entry. If you're collecting a lot of data, you can just upload in bulk. So this is about the data entry and, how you can collect the specimen data. As I said, these are all very generic configurations based on some of the demos we have done. But if you want to see something specific to your collections, your use cases, we would be happy to configure that on the demo site and give you access and do one-on-one demos as well. So that's about the data entry. So now let's go into the reporting module, so which is the queries module. Here you can get the data out easily. You can create your own reports, or you can access any of the default queries. So when you install OpenSpecimen, again, there are some canned reports that you can use by default for getting the data out, or you can create a new query. So here, when I'm creating a new query, I can either query for specific protocol, or I can query across protocols. So this is again dependent on what access I have. Then I can look for specific samples based on filters. So now I'll say, okay, give me all specimens for only alive patients, which is the vital status, only female patients, which is the gender. So this way I can add such filters on any field of OpenSpecimen, including any form fields that you've created or form fields that you've pulled from different systems. So I was talking about integrations with RedCap, Epic, and so on. Why is that required? Because you will then be able to do reporting based on those fields. Now you will be able to create reports across specimens and patients and clinical data and so on. So now I can say, okay, give me all specimens of specific type. So I can add again conditions on that. So I can say plasma, serum. So now what this means is, give me all patients who are alive, who are female gender, and give me all plasmas and serums for those patients. Now, like when I say view records, it will bring those patients with all the filters that I added. It will get me all the samples related data for that. So by default, it will show me some columns, but I can include any columns I want. So suppose I don't want any visit columns. I want, say, where they are stored. Suppose I want to see the container details. I can include that as well. So I can add any filter, like when I'm searching for the specimens, I can add filters and search for specimens. And I can also include any columns I want in the results view, so that there is no restriction on what field you filter on, what field you include in the results view. So if you see now, it's telling me which box it is stored, which location, which freezer, all the details I need, I can export it. I can save these reports so that I don't have to recreate this again. I can export such reports into CSV files. And I'll be also able to schedule reports. Like if you want to run an automatic report every month, I can do that. You can do a lot of complex queries. Suppose you want to see matching normal and tumor tissue specimens from the same patient. You will be able to do that. So for example, as a researcher, I want to see all patients who have normal as well as tumor tissues from the same patient. So if you see here, now this is the patient ID, these are the specimens. So these kind of queries can also be done. You can also do count-based reports, like if you want to just see summary of data, like specimens by different types and studies. As an admin, I want to see on a monthly basis just a summary of all the information. Then I can do such summary counts or pivot tables like Excel sheets. You can create them within OpenSpecimen. And all of these reports, you can build it on your own without any IT support. And you can, with the reporting module, you can also build dashboards like this, where you can have different graphs, like based on anatomic side, based on different types of specimen. So you can build such dashboards as well, which is easy to display on your biobank website, where researcher can get an overview of what you have. So that was the reporting module. And you also have a catalog module. If you want to publish your specimens to outside researchers, they will be able to look for specimens and request for specimens. So imagine whenever a requester or researcher wants a specimen, right now they might be calling you up asking for, okay, do you have specimens from female gender between age group of 10 to 50 with certain diagnosis? So now you have to go through your data and look for specimens. Instead, you can publish your data on this catalog using these graphs or even this catalog module, where the researcher themselves can filter on the data and look for the specimens. So now they'll say, okay, between age group of 10 to 50, I want the specimens and I need this only for female gender patients. So they can keep adding such filters, and they can see the list view of the specimens like this. They can again add conditions on diagnosis, type of specimens. And now once they are interested in certain samples, what they can do is they can also see more details. So what they see in the catalog, what specimens they see, what fields they see, you can control, and so that you can only show, say, very minimal data to the researchers to the catalog. And now if I, as a requester or researcher, I'm interested in these specimens, then I can add to cart. Then from the cart, I can request the specimens. When I submit the request, the biobank managers or request managers, they get notified. You can make the researcher fill up certain form. This form is also configurable. Now, once the request is sent out, so if I go back here, as a admin, I will be then able to, review all the requests, approve a request. So suppose if you see, I can approve or reject a request. And after I approve a particular request, they get notified, and you will be able to then distribute the specimens and close the request. So this is how, the whole life cycle of the specimens can be captured. If you see now, for the specimens that are sent out or shipped out to the researchers, orders are created. So this will track who was the requester. Like if you see who was the requester, when was it requested, where did they receive it, how many specimens did they receive, and if you see the specimen, once the specimens are shipped out to a researcher, it will be distributed. The storage location within your freezer would be automatically freed up. Available quantity would be made 0, and there would be a distribution event. So this will track the specimens that are sent out. So you have an audit of everything that happened to the specimen, if it is moved from one location to another, shipped out of your freezers, disposed, or everything you can track about the specimen itself. Then you should be able to then do reporting based on it. So these are some of the basic modules. There are other modules like you can do supplies or inventory management. Suppose you want to track your tubes, reagents, kits. There is a separate module to track supplies for that. We also recently developed a workflows module where you can track your SOPs step by step. So you can have separate workflows for blood processing, tissue processing, VBMC processing. How this would be helpful is, instead of navigating through multiple pages in the application, you can have like step-by-step process as if you are looking at the SOP and doing every step. So you receive the tubes in the lab, you mark it as received, and you mark the quality. Then you dilute the material. Then there is fecal isolation, centrifuge it, count the PBMCs, that is the cell count, and make the aliquots and store them. So this is an example of a workflow, but you can have such multiple workflows which can be shared across protocols, and you can track every step of what happened in the processing so that and you can do reporting based on it. If there were any deviations, you can do it. So this way you can have multiple workflows, as I mentioned. And again, these are some examples, but you can have any number of such workflows which can be shared across different groups, different CPs. And what this will help you is ease of data entry. It will save your data entry time because you don't have to do, all these steps in multiple pages, multiple clicks. And finally, you collect high quality data because you have every time point of what happened to the specimen, so that you will be able to do reporting based on that. So the workflows module will help you track that. So these are some of the features. As I said, this was just an overview. If you need a specific demo based on your use cases, we'll be happy to do one-on-one demos. As you see, starting from specimen collection, whether it is longitudinal collection or general biobanking, you can collect every data about the sample, how was it processed, how was it stored, and how was it utilized. So that's about it for today's demonstration. We are happy to take any questions you have. Otherwise, thanks everyone for attending, and we'll be posting the video if you want to share it with your colleagues, and contact us if you have any questions or if you need any follow-up call. Thanks everyone.
I have a quick question for you.
Yeah, sure.
So with the longitudinal study, the pre-entry, if the value's less than what we originally were thinking, so if I created aliquots Originally was thinking 5, but actually created 4. On the screen, it showed the 4 that I created were green. The one I didn't was yellow. Is there a way to remove that 1?
Yeah, so it just shows it. So there is a setting to hide the pending after a few days or even after immediately after collection. So this is just to show you that, out of 5, you collected 4, but you can hide the pending ones. There is a setting to hide them.
Okay. Thank you.
Okay, any other questions? Okay, that's it then. Thanks everyone again for joining. We'll post again for the next webinar. Looking forward to hearing from you if you're interested in OpenSpecimen or any follow-up questions you have. Thank you. Bye-bye. Have a great day.
Thank you.